Hello. We'll talk today about the RNA synthesis, which is actually the transcription of RNA from DNA. And we'll describe the structure of RNA, different RNA, the transcription of eukaryotic genes, the difference between the eukaryotic and prokaryotic RNA, and as well we will differentiate between the eukaryotic and the prokaryotic transcription. As well, we'll talk about the RNA processing, and we'll describe how the DNA eukaryotic precursor messenger RNA are processed to form a functional messenger RNA. Sorry, I switch off my phone. Okay. So what is the central dogma? Actually, DNA is transcribed into RNA. It's a single stranded nucleic acid, which is soon translated into protein synthesis. So basically, the gene, which is the hereditary unit of DNA, contains all the information to specify the synthesis of functional RNA to build a protein. So basically, the 4-base language of DNA, which is AGCT, is transcribed into another 4-base language of RNA, which is AGCU. That after that is translated into another language that consists of majorly 20 amino acids that form the proteins. So let's start with the structure of RNA. The primary structure of RNA is very similar to that of DNA, with 2 exceptions. The ribose, the sugar, has a hydroxyl group on the 2nd position, as you can see from the figure, on the 2nd position. And the T in the DNA is replaced by a U or uracil in RNA. Why is that? Why it's ribose and not the deoxyribose? In addition, the presence of T, rather U in DNA, is important to long-term stability of DNA. So we can see here that the OH that exists on the carbon 2 prime of the ribose sugar makes RNA more chemically labile than DNA. So it's a kind of a fragile than the DNA. As well, this group, this hydroxyl group, provide a chemically reactive group that take part in RNA-mediated catalysis, so in which RNA is cleaved into mononucleotide in lab by alkaline solutions. So these inside the cells facilitate the broken down or the degradation of the RNA after it did its job to be recycled and used again for the transcription and for the synthesis of a new messenger RNA. Keep in mind that it is the carbon 2 prime. It's not the carbon 3 prime, because carbon 3 prime, as we'll see later, will participate in the formation of the single-stranded RNA and the phosphodiester bond. So like DNA, RNA is a long polypeptide that can be double-stranded, sometimes you can say double-stranded or single-stranded, linear or circular. Generally, there are 3 types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. The RNA can form hybrid helix composed of RNA and DNA strands, as we will see later on. RNA and RNA-DNA strands, a double helix, have a conformation, as we said before, like an A form of a DNA. Some RNA molecules have internal catalytic capacity, which these kinds of RNA are called ribozymes. They can catalyze self-splicing during the formation of the majority of the functional messenger RNA. In addition to messenger RNA, ribosomal RNA and transfer RNA genes, there are thousands of additional genes that are transcribed into non-protein coding RNAs. So that's mean that they are transcribed and not translated. That's why they are non-protein coding RNAs. Examples include small nuclear RNAs, snRNAs, small nucleolar RNAs, SNO RNAs, and large families, more than around 2,000 in humans, of short microRNAs, miRNAs. Unlike DNA, RNAs are single-stranded and exhibit a variety of conformation. So here we'll see the conformation. Of course, the primary structure is a single strand RNA. So the first secondary structure, it's called hairpin. So in case of hairpin, you can see that it can do a double strand, and the loop here is around from 5 to 10 ribonucleotides. In case this loop is larger, it will form another structure of a secondary structure, in which the loop is around from 11 to 100 ribonucleotide. And you can see the region that form the double strand region. In case of this one, in case you can see two structures, you can see stem, which is actually a double-stranded RNA, and a loop, which is around the nucleotide from 11 to 100 nucleotides single-stranded. And these two structures are the secondary structure, are types of secondary structure. There is another structure, a high level of complexity, is the tertiary structure, in which in the tertiary you can see it can form two double strands, and still it is one messenger, one RNA. In this case, you can see two stems, the green and the blue, and you can see two loops as well. In this conformation, it is generally called pseudo knot, because it's a false knot actually. So this is the secondary structure and the tertiary structure. So what about the transcription of messenger RNA and how the RNA is synthesized? Keep in mind that the blue strand here is a DNA template in which RNA will be built complementary to the base on this template strand. And keep in mind that synthesis of RNA always synthesized in the direction of 5', 3' by a very important enzyme, which is the RNA polymerase. So here again, it is the same direction used it is the same direction as we said in last lecture in the DNA replication. So in DNA replication, as well as transcription of RNA, occur in the direction, as you can see from the figure, in the direction from the 5' into the 3'. In this direction. So you can see here is the ribonucleotide, and here is the base is complementary to the base to the template, okay? And is moving, building up the RNA opposite to in the direction of the template strand. So initially, for adding a nucleotide in the chain of the RNA, the incoming ribonucleotide, either A, U, C, or G, of course, it consists of a base and ribose sugar and triphosphate, alpha phosphate, beta, and the gamma phosphate. So the incoming ribonucleotide initially binds complementary with the base on the template DNA, and starts the attack occur between the, as we just said, the attack between the 3' carbon and the alpha phosphate. So you can see the polymerization start here, and the high energy bond between the alpha and the beta, phosphates is replaced by a lower energy bond, which is actually the phosphoester bond that it will be formed. The byproduct will be a pyrophosphate group that, by the help of an enzyme of pyrophosphatase, will break the pyrophosphate into phosphate. So let's see generally what's happening, okay? So you can see here, this is a sketch for the DNA, okay? So RNA polymerase, as we said, it moves in the direction from 5' into 3', and it sits on the DNA. It needs a specific sequence to localize the gene needed to be transcribed. This specific sequence is called a promoter. So specific sequence on the DNA, and this promoter usually is upstream of the gene. So you can see here from the figure to understand what is numbers. So initially +1. So let's see. So the site on the DNA template at which RNA polymerase begin to do transcription of the gene, normally it's called +1. Then the downstream denotes the direction in which the template DNA strand is transcribed. So downstream means that the transcription will go this way, and you can see it's highlighting the coding sequence start from +1. So the increasing the positive number indicate the downstream sequence of the gene, while upstream is denoted by negative numbers and represent the opposite direction in which the RNA polymerase will not move in opposite direction. Always the RNA polymerase will move on the template in the direction of 5', 3'. So in this sketch, we are representing the lower strand as the template. So in case it is the lower strand as the template, so the RNA polymerase, when it will sit on this strand after melting the DNA, the RNA polymerase will move in this direction, which will be 5', 3'. So as we said, the nucleotide position in DNA sequence downstream of the start site are indicated by positive, while upstream are indicated by negative. And here you can see as well, where is the location of the promoter region? Usually in the upstream of the gene. So generally the RNA polymerase need to localize this sequence of the promoter and sits in order to start transcription. It's like a guide for the RNA polymerase to pick the correct gene for transcription. And of course, because RNA is synthesized in the direction of 5', as I said, 3', so the RNA moves down the template in the direction, in the DNA strand, in the direction of 3', 5' of the DNA template. As you can see, we consider the lower one is a template. So you can see here is the template and here is the non-template. And this one is transcribed into primary RNA transcript. So why we need to call it a primary RNA transcript? Because it needs further processing in order to be a functional messenger RNA. The RNA synthesis, as we said, complementary to the template strand, and therefore it is actually identical with the non-template strand sequence, except with U in place of T. So basically you can understand from that actually if you have a double strand, one of them is template and one of them is non-template, basically the RNA that will be transcribed from the template, it will be the same as the non-template. Instead you replace T's with U's. As you can see here, this is the primary RNA transcript. It is actually the same as the non-template one, except that the T have been replaced into U. So let's start talking about the actual mechanism of transcription of RNA, and we'll start by the transcription of RNA in the prokaryotes. Okay, so generally it is divided into 3 stages: initiation, elongation, and termination. So let's start with initiation in prokaryotes. So here is the DNA, double-stranded DNA. Green here denotes the start site on the template DNA, which actually +1, and red represent the stop site on DNA. So the gene need to be transcribed starting from here to here, okay? So you can see that the RNA polymerase didn't sit on the gene, but sits on the upstream region of the gene, which is called promoter, In this case, this RNA polymerase required initiation factor, IF, initiation factor to recognize this promoter, okay? To recognize the region and the sequence of the DNA which is upstream of the specified gene need to be transcribed. In this structure is called the closed complex. Why it's called the closed complex? Because still DNA is not melted yet for the transcription. So again, this region of DNA sequence is called upstream, while this region is called downstream. And as I just told you, that the green represents the +1 or the initiation, the start site of the on the template for transcription. Melting start to occur. So melting, that's mean the hydrogen bonds are broken. This melting result into something is called transcription bubble. So you can see a bubble that open in the DNA double strand, and this bubble around 12 to 14 base pair. And in this case, this complex is now called open complex, not closed complex, because the DNA already melted at the region of the start site of transcription. So initially, the RNA polymerase, this whole yellow structure, the RNA polymerase start to add nucleotide complementary to the template strand. Complementary to the strand, and you can see this is represented by this red strand, red small strand, which you consider the initial ribonucleotide that have been added by the RNA polymerase on the template strand. And it is recognized that once the first two initial ribonucleotide is added, this mark the termination of the initiation process, because since the third one will be added, it will be a repetitive addition, so it will go into the elongation step. So let's talk about the elongation. So you can see here that the RNA polymerase has already moved. That's one major change. The second major change that you can see that the nascent RNA, the new RNA, the transcript have been formed and it is long. Part of the RNA is already loose and the other part of RNA is still attached or doing a RNA DNA hybrid double structure. Again, that's the RNA DNA hybrid double structure. So what about this RNA, nascent RNA? This DNA RNA hybrid structure is consisting of 8 nucleotides, and this is the hybrid region. And the melted region now, which moves along the gene until the stop site, will be of the size of 14 to 20 base pair. So normally the RNA polymerase melts the DNA and keep this region, melted region of around 14 to 20 base pair along its movement over the gene required to be transcribed. You can see as well that the free end of the nascent RNA is 5', and that proves that transcription occurs in the direction of 5' to 3'. So this end will normally be 3'. So one ribonucleotide during this elongation process, one ribonucleotide is added at a time at the free end of the nascent RNA, okay? The only thing that that is very recognizable here, that the presence of DNA RNA hybrid, which is around 8 nucleotide in length. That's elongation. Soon after elongation and until reaching to so once the RNA polymerase reach to the stop site or stop sequence of the DNA, it will do a termination, which is the last step. In termination, everything is released, the completed RNA strand, you can see it from the 5', 3', and the RNA polymerase waiting to do another round of transcription on the same gene or on different gene. So RNA polymerase actually can transcribe very long genes up to 2 million base pair without dissociating from the DNA template or releasing the nascent RNA. And this is very crucial. This stability is very crucial because sometimes our body needs a gene to be transcribed, and if the association of the RNA polymerase is not very strong with the template, it will cause a lot of problems. RNA synthesis occur at a rate of around 1,000 to 1,000 to, let's have a pointer, 1,000 to 2,000 ribonucleotide per minute at 37 degrees. So it's around average of 1,500 ribonucleotide per minute, and that's a pretty quick comparatively. So let's continue to talk about prokaryotes. So we just said the general mechanism of the prokaryotic transcription. So how the organization of genes differs in prokaryotes and eukaryotic DNA. Prokaryotic DNA, genes actually are closely packed with very few non-coding gaps. So what about operon? So operon actually operates as a unit which under the control of a single promoter. So actually operon could be a number of genes under the control of a single promoter. As we said before, promoter is the important sequence which is upstream of the gene and important for recognizing of the RNA polymerase to the gene needed to be transcribed. So this one of the major difference between the organization of genes between prokaryotes and eukaryotes, in which there is an operon in prokaryotes, in which you can see a multiple number of genes can be under the control of a single promoter, while in eukaryotes, each gene has its own promoter. So the operon is transcribed into continuous strands of messenger RNA that carries the message for a related series of protein. So that's mean a related series of proteins, different proteins. So that's mean there were initially different genes. Each section of messenger RNA represents a unit or a gene that encode one protein in the series of these proteins. But eukaryotic DNA encoding genes that function together are often separated in DNA. Each gene is transcribed from its own promoter, producing one messenger RNA. So the nucleotide sequence of eukaryotic messenger RNA revealed that eukaryotic genes exist in pieces of coding sequence called exons, separating by non-protein coding segments called introns. The conclusion at that point that the long initial primary transcript messenger RNA, which is the nascent RNA, had to be clipped apart in eukaryotes to remove the non-coding sequence, which is the intron, and then stitch back together the exons to produce a functional messenger RNA. This is one of the processes that occur in RNA processing. So introns are common in multicellular organisms, the non-coding regions, but extremely rare in bacteria and archaea, and uncommon in many unicellular eukaryotes such as yeast. So let's see an example here about the tryptophan operon. So this example describes how the synthesis of the amino acid tryptophan occur in prokaryotes and eukaryotes. So in prokaryote, you can see that the tryptophan operon consists of 5 structural genes, which is termed EDCBA, trap A, trap B, C, D, E. These 5 genes have only one start site on the DNA, that's the E. coli genome, the gene responsible for the tryptophan. Tryptophan is called the operon, responsible for the tryptophan synthesis. So you can see it's having only one start site. Of course, it will need one promoter for the RNA polymerase to sit on this promoter and to transcribe all these series of genes. While in the case of the eukaryotes, it's under the control of the 5 differently localized genes, TREB 1, 2, 3, 4, 5. And you can see that all genes are largely separated, even located on a different chromosomes in the yeast. And yeast is one of the example of the simple eukaryotes. So you can see that TREB 1 and 4 localized in chromosome 4, while TREB 2 on chromosome 5, TREB 5 on chromosome 7, and TREB 3 on chromosome 11. So this shows how the gene is organized differently between the two types of organisms. In case of transcription, you'll see in prokaryotes, you will have only one single nascent RNA, one single primary messenger RNA, one single transcript of RNA. But in case of the eukaryotes, we'll have 5 separated messenger RNA. Each of them have generated from under the control of different promoter. But here the single-stranded RNA, just one, have it all generated from a single promoter. And soon after that, they will be translated, and finally you'll have 5 different proteins. Here as well, 5 different proteins. So you can see how it's the gene, the organization, although the genes are organized differently in prokaryotes and eukaryotes, but still will give rise to 5 different proteins. So what is controlling the eukaryotic RNA polymerase this function? All eukaryotic cells contain 3 different RNA polymerases, pol 1, 2, 3, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Each eukaryotic RNA polymerase transcribes genes encoding different RNAs. So you can see that RNA polymerase 1 actually it is localized in the nucleolus, and it is responsible for the formation of the ribosomal RNA, which function in the ribosomal components and protein synthesis. While RNA polymerase 2, it is localized in the nucleus and majorly concerned with the transcription of messenger RNA, and of course that is implicated in the formation of a protein. As well, it deals with other non-coding RNAs. For RNA polymerase 3, it is localized as well in the nucleus, and it is majorly concerned with the transfer RNA, which have a major function in the protein synthesis as well. And as well, it is involved in the formation of some of the RNAs that share its part in the ribosome component, and as well other non-coding RNAs. So let's see. So let's see how the transcription factor position RNA polymerase 2 at the start site and assist in the initiation in eukaryotic transcription. So we'll talk now about the eukaryotic transcription, and we'll start with the initiation stage of the eukaryotic transcription. So initially, you have a DNA sequence, and of course, there's a TATA box. In this part of the DNA sequence, actually, it is rich in T and A. That's why it's called TATA box. So this TATA box represented the promoter region of the gene needed to be transcribed. So you need to understand that upstream of this box, it will be the gene need to be transcribed. Here, the transcription factors actually that position this polymerase 2 on the promoter region in order to do the transcription. Actually, there are a number of general transcription factors. So they take letters. So you can see here the first transcription factor. The first transcription factor is called TF2D. So TF2D, it's a transcription factor 2, and the 2 is related because it is working. These transcription factors is working with polymerase 2, and as we said before, there are 3 kinds of polymerase. So these transcription factors are related to polymerase 2. And D, of course, it's a letter to differentiate between the different transcription factor. So here, so what about the TF2D? So TF2D, actually, it's a TATA box binding protein and 13 TAFs, TPP associated protein. So basically, it consists of 2 major classes of protein. The TPP binding, the TPP protein here, the blue one, which is the TATA box binding protein, and the 13 TAFs, which is the TPP associated factors. So this is a transcription factor 2D. So what is function? Actually, the TPP is the first protein to bind to the TATA box promoter. So this TPP interact with the minor groove in the DNA and leading to bending of the DNA. Soon after that as well, the TAFs, around 13 TAFs, function in initiating the transcription from the promoter that lack TATA box. So it's a kind of another mechanism to sit on the promoter that lack. There is another sets of promoter that lack Tata box. In these cases, TAFs come in action. Soon after that, you can see that once TF2D recognize a promoter, the TF2D has bound TF2A, TF2A and TF2B can bind as well. You can see it bound, and you can see that the DNA is already bound now. Okay, and you can see the +1, which represent the start site of transcription of the gene. And in this case, these complexes, now it's called upstream promoter complex because it's already bound to the promoter and it is still upstream of the gene. So the TF2B bind to the major groove of the DNA and assist the polymerase II in melting the DNA strand at the transcription start site. So we understand. So we can understand now that TF2B have a melting capacity or helicase capacity. Soon after that, the TF2F binds to polymerase II, comes in action now, and helps in positioning the polymerase over the start site. And now the complex is called core PIC. So what's core PIC? It's a core pre-initiation complex. So the complex of polymerase II now with the general transcription factor, which is TF2F, TF2B, TF2A, TF2D, so it's a 4 transcription factor, all together bound to the promoter and ready to initiate transcription. And again, this complex, which is still didn't move yet, it's called core PIC, core pre-initiation complex. Again, the core PIC, as you can see, this is the core PIC, didn't move yet to the gene. It's another 2 transcription factor, which is TF2E and the TF2H incorporated into the complex. For TF2E, it creates a docking site for TF2H. So TF2H is very important in the process of eukaryotic transcription, as we will see later. So it needs to be recruited nearly at the end before initiation of the transcription. And it needs a specific transcription factor here, it is the TF2E. So TF, as we said, TF2E recruits the TF2H. As you can see, TF2H have 2 different protein subunits. Okay, so you can see here that the complex already bound. So the binding of TF2H complete the assembly of the PIC. So now it's called closed PIC, closed pre-initiation complex. And again, why it's closed? Because still DNA is not melted yet. So the PIC assemble at the promoter of every protein coding gene expressed by a eukaryotic cell. By the help of ATP, you can see that it will start to open up the DNA, and now the complex is converted from closed PIC into open PIC. So how the DNA is melted? Actually, the helicase activity of TF2H unwind the DNA at the start site, allowing the polymerase to form an open PIC. This DNA duplex surrounding the start site is melted. And again, a transcription bubble will be visible. You can see the DNA melted at this region. And now the RNA polymerase are bound to the active site. And is ready, so the whole complex, the open PIC complex, is now ready to start its first move for transcription. So this, again, this open complex, as the polymerase transcribe away from the promoter region. So it's moving in the direction of 5', 3' with respect to the RNA that is formed. So as it moves away, TF2B is released from the RNA exit channel as the 5' end of the nascent RNA enters it because it starts to emerge. You can see here that TF2H, the kinase activity, So we have, as I said before, the TF2H consists of two subunits. One of them is a helicase activity and participate in opening up of the DNA, while the other part is a very important part that have a very important job, which is a kinase activity. So what's a kinase activity? Kinase activity, that's mean it have the ability to phosphorylate. So this subunit of the TF2H phosphorylates the carboxyl terminal of polymerase 2, which is denoted here CTD. So this carboxyl terminal domain of polymerase 2, the phosphorylation of this domain is crucial for the function of RNA polymerase. Once we reach this complex, it's now converted from an open complex into initially transcribing complex. It's ready, and why it's initially transcribed complex? Because the RNA polymerase start to emerge, and you can see the 5' end here of the nascent RNA. So here you can see that the kinase activity of the TF2H have phosphorylated the CTD domain of the RNA polymerase. The energy released from the process of phosphorylation is very important in providing the energy needed for the RNA polymerase to move along the DNA. And you can see here how the DNA is melted, the RNA polymerase, and the emerging of the 5' end of the messenger RNA. Okay. And now the complex doesn't need anymore the transcription factors, which is actually called initiation factor, because these transcription factors participated in the initiation process, and now it needs another set of factors, is the elongation factors. So initiation factors is released, and a new set of elongation factor is introduced. And now, as I've said, the complex now is called elongation complex. So polymerase 2 initiate transcription in the resulting open complex, and the polymerase transcribes away from the promoter. So its CTD domain, CTD becomes, as we said, phosphorylated by TF2H, kinase domain, and as well the general transcription factor dissociate from the promoter, leaving the RNA polymerase for continuing the elongation phase. Okay, as you can see here, so what this figure represent? So actually this is an electron micrograph, real electron micrograph showing the process of transcription. So to understand, the green arrow shows DNA strand, double-stranded DNA. This is double-stranded DNA in the region that it is not transcribing, not actively transcribing. While all this structure that appear here, it is the region that actively being transcribed. Every single hair-like structure like this, every single hair-like structure like this is representing one messenger RNA. So you can see that every single gene is transcribed, and it's only not transcribing one copy of the RNA. It can transcribe many copies of RNA, okay? So this region of actively transcribing gene, and this is a region which is represented by the red arrow, and this is a region of inactively transcribed gene, or maybe it's a non-coding region of a DNA. So now we'll talk about the second subtopic of the lecture, which is RNA processing, and we'll describe how the DNA eukaryotic precursor messenger RNA, which we call it a nascent RNA or primary transcript, are processed to form a functional messenger RNA. So in eukaryotes, these precursor messenger RNA are processed. In bacterial, in prokaryotes, the translation of a messenger RNA into protein can begin at the 5' end of the messenger RNA, even while the 3' end is still being formed by RNA polymerase. As we said before in our last lecture, so transcription and translation occur concurrently in bacteria, and of course this is due to the lack of the nuclear envelope. But the case is different in the eukaryotes. In eukaryotes, the primary transcript or precursor messenger RNA or pre-messenger RNA or nascent messenger RNA or heterogeneous messenger RNA. All these names actually the same, needs to be processed. The RNA needs to be processed to have a function messenger RNA. So how is processing occur? Actually, RNA processing, basically it's a 3 modification for the nascent RNA. The first modification of this in the RNA processing is the addition of a 7-methyl guanylate to the 5' end of the nascent RNA. This 7, you can see here, 7, this is a nucleotide, 7 methyl, because there is a methyl group on the nitrogen number 7, guanylate, because basically it's a guanylene. So this 7-methyl guanylate is added to the 5, as you can see, 5' end of the nascent RNA. Normally, for simplicity, the 7-methyl guanylate can be written 5' cap, because it is at the initial nucleotide of the messenger RNA, or we can say it M7G triple P. Why triple P? Because this 7-methyl guanylate is linked by a very strong linkage, very specific, very peculiar linkage, which is called 5' linkage. 5' triphosphate. You can see 3 phosphate groups, triphosphate linkage. So why messenger RNA, this nascent messenger RNA need a cap? Before explaining the function of the cap, we need to understand that additional structure occur that share its part in the capping process is the having the methylation of the hydroxyl group of the first and the second group. Actually, we are blocking down the activity of this hydroxyl group, as you can see, because basically they are OH, because it's a ribose. Okay, so the methylation here for this first base and second base will block the activity of this functional group at the first 2 nucleotides. So what is the function of the cap? Actually, the cap, which is represented by the 7-methyl guanylate, protect actually messenger RNA from enzymatic degradation, and helps as well the messenger RNA to be exported from the nucleus into the cytoplasm. As well, the cap can be bound by a protein factor that is required for being translated in the cytoplasm. So actually it has 3 different functions on 3 different levels. So it helps, this cap help the messenger RNA to be recruited to the cytoplasm, help the RNA to not to be degraded, and help the RNA to be translated, okay? So what about the other 2 modifications that occur in the processing? So you can see here, this is represent like a nascent RNA with a 5' end and 3' end. So the second modification is the addition of a polyadenine tail at the 3' end. We have just talked about the capping that's happening on the 5' end. What about the addition of the polyadenine tail? It starts with cleavage by an endonuclease. Endonuclease, what's the meaning of endonuclease? Endonuclease, it's an enzyme that work on nucleic acid, and it hydrolyzes the nucleic acid internally from the structure internally, not from the terminal sites. So the cleavage of the endonuclease in this phosphate group will yield an OH group. So basically at the 3' end, at the last nucleotide of the nascent RNA, the phosphate group is converted to an OH group. So why it's converted to an OH group? To make it a reactive group again. So after conversion into OH group, an enzyme which is called the polymerase adds a string of adenylate acid residues that can be reached to 100 to 250 bases. Okay, so adenine is added to this end. So basically, the RNA now will have a cap at the 5' end and the polyadenine at the 3' end. And this polyadenine will can reach to up to 250 bases of adenine. So what's the function of this polyadenine tail? Of course, it is a stabilization and very important as well in translation of messenger RNA. What about the third modification of RNA processing? It's called RNA splicing. And actually RNA splicing, it is the internal cleavage of the transcript to remove the introns and stitch back together the coding exons. So actually the functional eukaryotic and prokaryotic messenger RNA have as well non-coding region. So you can see here. Additionally, this non-coding region at each end, which is called UTRs. So in both ends, in prokaryotes and eukaryotes, have a non-coding regions at the 5' end and 3' end, which is called untranslated regions, and normally it's denoted as UTRs. These UTRs participate in the regulation of messenger RNA translation and stability. So generally, these are the three modifications that occur for the nascent RNA in order to be a functional RNA. To summarize these three modifications in the RNA processing, you have the capping process, methyl 7-guanylate, you have an polyadenine tail, and you have the third one is the RNA splicing, which is the removal of the introns and stitching back of the exons. So let's see an example of how it occurs, the whole process. So in this example, you can see this is the gene of the beta globin genomic DNA. The red regions represent the exons, while the blue regions represent the introns. As we said, at the 5' and the 3', you can see the position in the DNA sequence of the untranslated region. Here is the start site of RNA synthesis, and here is the end site. And as well, this is the site of the polyadenine tail. The numbers here denote the sequences of the introns and the position of the nucleotide of the introns and the exons. It starts with the transcription. So you can see here the transcription gave rise to a messenger RNA that is not completely functional. You can see there is a cap here. This is a methyl, the green circle. It's a methyl 7-guanylate triphosphate cap. Exists at the 5' end. You can see still why it's still not functional, because still you can see the introns and the exons, and as you can see the gray regions is the UTRs. It needs a process of cleavage of the phosphate and the conversion to OH, and the addition of a polyadenine tail, which we said that it's around from 100 to 250 nucleotide according to species. So it needs the third processing modification, which is the removal of the introns. So here the intron is removed, and finally you have a functional messenger RNA from 1 to 147 nucleotides from 1. So the size of this RNA is 147 nucleotides, have a cap at the 5' end, have a polyadenine tail at the 3' end, and have 2 UTRs at the 5' and 3'. And this is the functional beta globin messenger RNA. Let's see why this splicing, why actually there is introns and exons. Why not in eukaryotes we have only coding regions? Let's see an example, very important example, that there is a process which is called alternative RNA splicing. The alternative RNA splicing increase actually the number of protein expressed and give another layer of diversity of the protein that have been coded from the DNA. So in contrast to bacteria and archaeal genes, the vast majority of genes in multicellular contain multiple introns. So introns are a characteristic feature of eukaryotic DNA. The presence of these multiple introns in eukaryotic genes permits the expression of a multiple related protein from a single gene. That's another layer of expression of genes by means of alternative splicing. In higher eukaryotes, the alternative splicing is an important mechanism for production different forms of protein. So it's not totally different forms of protein. So that's why we call it isoforms. by different types of cells. So that means you have the same genes in two different types of cells. But although you have the same genes, due to alternative splicing, you can see that you have two different isoforms of the protein produced from that genes due to alternative splicing, as you will see in the next few slides. Let's take, for example, fibronectin. So what's fibronectin? Fibronectin, actually, it's a multi-domain extracellular adhesive protein found in mammals. And it is secreted by two, for our example, you can see it is secreted by two different types of cells, fibroblasts and hepatocytes. And we will explain a very good example of alternative splicing and see how this cell, in eukaryotic cell, there is another layer of expression of genes, gives another layer of complexity and understanding to the way to manipulate the need of the function of each cell differently by expressing a protein that relates to its function. So for example, in fibroblast, this fibronectin messenger RNA contain two exons called E3A, E3B. These two exons in fibronectin encode a protein domain that enable the fibroblast to adhere to extracellular matrix. Again, so the protein version, the isoform version of the fibronectin formed in the fibroblast have two exons, and these two exons enable that protein isoform to be sticky to the extracellular matrix, while in hepatocytes, these fibronectin lack these exons. So actually the fibronectin which is produced in the hepatocyte will lack this sticky behavior, because it will be allowed to, which allows it to circulate, okay? So that's mean it will not help in the sticky behavior. Okay, let's see again in diagrams. Generally, fibronectin have more than 20 different isoforms. We only talk about two isoforms, the one in the hepatocyte, the one in the fibroblast. So it has generally 20 different isoforms of fibronectin have been identified, each encoded by different alternatively spliced messenger RNA composed of a unique combination of the exons from the single gene, which is the fibronectin gene. So again, so you have, for simplicity, if you have of course, the gene of fibronectin is existing in all of our cells. So if we have like 20 different type of cell, these fibronectin have a 20 different version of proteins of fibronectin, which is called the isoform, to suit the function of each different cells. So let's see diagrammatically what's happening. So here is the fibronectin gene. So the boxes, all the boxes are the exons. The black lines, the black double line, is the regions of the entrance. So you can see here, we highlighted these, again, all these boxes with any color is the exon, while the black lines is the entrance. And you can see here, E3A and E3P, the two sequences of DNA, the two exons on the DNA that will, upon transcription and the translation, will translate it into specific sequence in the protein fibronectin that will be responsible for the sticky behavior of the fibronectin protein. So what's happening in fibroblast? Again, the DNA is transcribed into a fibroblast fibronectin messenger RNA, and you can see this functional messenger RNA in the fibroblast is having the two exons. So these two exons, as we said, encode the amino acid sequence that help a fibroblast to adhere or stick to extracellular matrix, while in hepatocyte, these two exons have been excised, have been removed in the process of alternative splicing. So basically, this hepatocyte version of the fibronectin messenger RNA will be translated into a fibronectin protein in the hepatocyte that lacks the sticky behavior of the fibroblast. So this is very obvious, actually, because if it expands our understanding of how the gene is manipulated inside each cell, because generally hepatocyte is sessile cell. It's not freely circulating cells, but fibroblast is freely circulating cells, and it circulates in the blood. So it needs to have a protein that help these type of cells, the fibroblast, to stick in order to not to be flushed away by the bloodstream. But in case of hepatocyte, it exists in the liver. It's not a moving cell, so it doesn't need this sticky behavior in the fibronectin. Okay. So now we'll talk about the transfer RNA, the second type of RNAs. We have just dealt with the messenger RNA. So transfer RNA are 70 to 80 nucleotide long. In bacterial cells, they are around 30 to 40 transfer RNAs, while in animal cells, it reaches up to 100 RNAs. So basically, the structure of the RNA in solution, all transfer RNA fold into stem and loop arrangement that resemble a clover leaf. So you can see stem, 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 loops. It have like 4 base paired stem, as you can see from the figure, and it have 3 loops, 1, 2, 3 major loops, 1, 2, 3. It have D loop, it have T epsilon C G loop, it have the anticodon loop. And of course, the base pairing, you can see double-stranded, double-stranded parts of the RNA, and that's RNA double strand. One of the peculiar characteristics of the transfer RNA is the existence at the 3' end, the existence of a CCA sequence, which exists on all different types of transfer RNA. And of course, you know, the main function of transfer RNA is recruitment of the amino acid, because amino acid binds to this end at the 3'. Several bases in most RNA are modified after the transcription of this transfer RNA, and they play an important role in protein synthesis. Of course, the transfer RNA is considered to be a non-coding RNA because transfer RNA is not translated. Viewed in 3 dimension, transfer RNA appear as a L shape. So you can see in 3D dimension, you can see the loops, and you can see the CCA at the 3' end. You can see the anticodon loop, okay? So how it works, the transfer RNA? Actually, the amino acid need to be linked to the transfer RNA at the 3' end, at the CCA sequence. So to participate in protein synthesis, this transfer RNA must be chemically linked to a specific amino acid. The linkage is catalyzed by a specific enzyme called amino acyl transfer RNA synthase, and cells have 20 different one of them, of these synthetases. So the amino acid is linked to the transfer RNA by a high energy bond, and hence once the transfer RNA is bound to its specific amino acid, it is called activated transfer RNA. The energy of this bond, the high energy bond, is used for the formation of the peptide bond, which will link the adjacent amino acid in the growing polypeptide chain. We will talk about this in details in next lecture about the protein synthesis. Because amino acids are so similar in structure, these synthetases can do mistakes. Okay, let's see how these amino acid transfer RNA can do mistake and they can repair these mistakes. So mistakes are corrected by the enzyme itself. The error rate of translation translation in E. coli is very low. It's around 1 to 50,000 codons. So you can see here, this is amino acid transfer synthetase, which is specific for the amino acid phenylalanine. Actually, this enzyme, again, the main function of this enzyme is to link the transfer RNA with its specific amino acid, which make it obvious that we need at least 20 different of these version of this enzyme, because generally we have 20 different amino acids. So generally these amino acid transfer RNA have two sites, and we'll talk about the one which is specific to phenylalanine. So one site for the attachment of the amino acid phenylalanine, the other site for attachment of the transfer RNA which is specific to this amino acid. And you can see here at the anticode loop, you can see that it have the codon that will give rise to phenylalanine. So it's still unbound the transfer RNA to the amino acid. So with the use of ATP, the linkage occur between the transfer RNA and the amino acid through, as we said, the high energy ester bond. And in this case, it's called amino acyl transfer RNA. And as we said before, it's called activated transfer RNA. Okay, so again, this transfer RNA recruit this amino acid to the ribosome to sit over the messenger RNA with the complementary code to have the amino acid in action for the polymerization for the amino acid for the protein. So now the phenylalanine is selected by its codon, which exists on the messenger RNA to be inserted in the forming of the protein chain. So this concludes our lecture, and you can see the reference for DNA synthesis, the last lecture as well again, and the reference for the RNA synthesis, transcription, and the processing, chapter 5 and chapter 9 with pages. Thank you, and I'm waiting for your questions.